We're going to bring up now two other scientists to join us in a discussion, and I wanted to introduce them and to have all of our speakers as well as those joining us in the discussion join us now at the table here. Dr. Susan Ross, who is, there she is the Measy Professor of Microbiology and Associate Dean and Director of Biomedical Graduate Studies at Penn School of Medicine. She studies the genetics of host-virus interactions, particularly retroviruses and New World arenaviruses. She's received several awards, a number of them from the American Society of Microbiology, and she was elected a Fellow of the American Academy of Microbiology in 2002. And I also want to introduce Dr. Theodore Schurr, who's studied the genetic prehistory of Asia and the Americas through studies of mitochondrial DNA, Y chromosome DNA, as well as autosomal DNA variation in Asian, Siberian, Native American populations. Right now, he's studying genetic diversity in Native American populations in the US and Canada, as well as indigenous Siberian groups from the Altai-Sayan region, if I'm pronouncing that correctly, and archaeological populations from the Lake Baikal region. He's also here at Penn in the Department of Anthropology, as well as at the University of Pennsylvania Museum of Archaeology and Anthropology. A number of you, Dr. Bushman, you were talking about diet, and you actually fed them various meals and things. And Dr. Tishkoff, you actually were looking in African populations at different characteristics. So you're kind of both looking at outcomes, if you will, one of which is a more or less direct outcome of genetics plus environment. The other one, you're kind of playing with the environment a little bit more. And the question from the audience was, how do you actually account for those differences when you're studying genomics, when you're studying genetics? How do you account for whether it's diet, where here, obviously, Dr. Bushman, you're doing a study. How do you look at diet or lifestyle in the populations that you study? How do you account for those differences when you're looking at genomics?
Well, that's a very good point. Almost certainly the host genotype, the human DNA, and environmental things like what you eat are influencing the composition of the gut microbiome, and then the outcome of whatever the microbiome is on a disease state or health or what have you. So those are two variables that are very much wrapped up together. We spend a lot of time scratching our heads on about how to study that. And so there's really no solution except to get a lot of data to study people with different genotypes, people on different diets, and get lots and lots of samples, and then try to do a bunch of mathy business to understand which are the components that are having the strongest effect on the microbiome composition or the disease outcome or what have you. So they're wrapped up together, and it's a considerable complication in our studies.
So the way that we try to do this, one of the ways is that we would look at, say, two populations that have a similar genetic ancestry. Maybe they had a common ancestor within the past 5,000 years, but they have a very different diet. And just one example that comes to mind is two groups we're studying in Kenya, one of them called the El Molo. They both originated from southern Ethiopia. And the group called the El Molo lives along Lake Turkana, and their diet is predominantly fish. And then the other group went further south to southern Kenya, and they were foragers and had actually a lot of honey, and recently have started having some cattle products. And so we can essentially control for a shared genetic ancestry and then look at what is the effect of having a different diet on some of these traits. But I just wanted to mention that in the future, we're hoping to, maybe I can team up with Dr. Bushman here, because we would love to collect poop, a little bit more challenging, actually, probably than even collecting blood, and looking at how these traits have co-evolved. So for example, I'm really curious to know, in the pastoralist groups, and some of these groups that have cattle, are there bacteria that have co-evolved together with the genetic variants and with the practice of cattle domestication?
There actually was a recent paper comparing gut bacteria in African kids and European kids, and they were found to be distinguishable. So something to study there.
I want to actually follow up because there were a couple of questions from the audience about lactose intolerance. And the first question is sort of the fundamental one, is what did you hope to learn from that? What can you learn from that? And the second is actually about whether or not domesticated cattle, has that had any role, if so, in any way, whether it's because of the microbes or because of something else happening?
I'm not sure I totally got the second question, but I'll start with the first. I was interested in studying the genetic basis of lactose tolerance since I did research in South Africa in 1997. And it was just a trait, a classic trait that was thought by anthropologists to be a classic example of an adaptive trait. And at the time, we knew nothing about the genetic basis of adaptation to consuming large amounts of milk. And it wasn't until 2002 that a group in Finland, where it's considered just a terrible thing if you can't digest milk, because almost everybody in the Finnish population can, and they found a genetic variant that's associated with the trait. So we looked in our Africans, but they didn't have it. And we were very curious to find out, well, if they don't have that, there must be something else. And so we actually went into the field and gave the sugar lactose and looked at the how well they were able to digest it, and were able to find these variants that turned out to be in a similar location, but were distinct from the Europeans. And it was a great example also of what we call convergent evolution, sort of multiple ways of coming up with a common solution, I guess you could think of it. And also it's of interest to NIH. I actually just went to a meeting in the last few months at NIH, which had a lot of medical doctors, people from the dairy industry, because for many, many years, many people in the medical community did not realize that the majority of the world can't digest milk. The ancestral state is to not be able to digest milk, and only in those of us whose ancestors had cattle and practiced dairying we're the ones who are able to, who have this adaptation. And so it's really important, that could play a role in diagnoses, but also in terms of policies and giving out powdered milk to children in developing areas and so on.
Thank you. I think the second, you've actually sort of answered the second part of the question, which is, and maybe the follow-on to that is, Dr. Bushman or anyone, did any part of that evolution take place because of microbes, either joining forces, so to speak, either on the cattle side, the human side, somewhere else?
That's a great question. We should go study the microbes of the cattle and
we'll study
the
that
humans
as well then. I think we might even find more ways to fund it.
I don't think we know, but we do know there is a correlation between, we can study the evolution of the cattle and sort of when they were introduced into Africa and other regions, and that does appear to be correlated with the genetic history in humans. So there is that level of correlation.
This is a point that Jim mentioned at the very beginning, that as Dr. Tishkoff just said, one of the lessons from genomics is, in fact, many of the things that we saw might be caused by or influenced by a single gene. Actually, it really depends on the whole genome. So in certain populations, it's a different gene, or actually a whole suite of genes that can influence a particular complex trait. And things also that we saw were simple, such as lactose intolerance, can turn out to be multigenic, so that means multiple factors in our genome and a complex trait, and that's really one of the lessons from the genomics.
Another question that I think is sort of a fundamental problem, if you will, and Dr. Bushman, maybe you're well placed to answer this, but is there anything about the genomes of various bacteria, viruses, et cetera, that can distinguish whether they are, in fact, I think as you put it, helpful, harmful, or just hanging out?
Sure. There's very extensive literature on that. Some viruses encode toxins that are directly harmful to tissue. There's a whole literature of virulence factors. Bacteria are amazing in that they're extremely plastic. So you can have very closely related bacteria, some of whom have a complement of virulence factors and are really bad, others of whom are very closely related, except they don't have the virulence factors and they're not bad at all. So it makes it hard to study these kinds of populations because you really need a lot of information to know if those genes are there or not. So yeah, to a very considerable degree, the sort of naughty genes are very recognizable in these kinds of sequence sets that we have. And so it does allow us to ask a lot of questions about adverse communities or the presence of pathogenic bacteria or viruses and so forth.
And Dr. Schurr, this may be one for you, and please, anyone else as well. Some of the genetics that we're looking at are actually things that we wouldn't think of off the top of our heads as good things. I'm thinking in particular, say, sickle cell anemia, which, as I'm sure many in the audience know, is actually potentially protective, and in fact has been shown to be protective of certain diseases, malaria in particular. How does that actually work? Not so much that, but how does the evolution of that work in various populations, and what is the selection process, if that's in fact what's happening?
This is a question that Sarah can also address, since a lot of her work has been directed towards understanding the adaptations to malaria. But in essence, we're dealing with what are called balancing polymorphisms, where mutations which would otherwise be deleterious in the case of the sickle cell trait causing anemia wind up having protective effects against the plasmodium that infects our red blood cells, because it confers a certain amount of resistance to infection by those particular parasites due to the sickling of the red blood cells. And so persons who are somewhat anemic actually have greater adaptation to or are better situated to.
When you say anemic, I'm sorry to interrupt, you actually mean just a low blood count?
Yes, low blood count, but the effects of anemia themselves, of it itself. So they're slightly unhealthy, but they're healthier than people who don't have sickle cell traits, because they are more susceptible to malaria and have lower fitness in malarial conditions for normal copies of the beta hemoglobin gene. So these kinds of balancing polymorphisms, there are others that we know about, but the sickle cell trait is a classic example. And actually there are a variety of different kinds of variants, again, some of which Sarah has been involved in characterizing, like glucose 6-phosphate dehydrogenase, which have occurred multiple different times because of the significance of the malarial pressure on human populations. As Sarah herself mentioned, that there are, I think it was mentioned before, there are different ways to get to the same point. And so this is something that we know about through, especially through these polymorphisms that affect hemoglobin. But there are others across the genome that we're becoming more and more aware of, these kinds of balancing polymorphisms.
So I think we're going to find out more and more that the infectious diseases in a population probably select for these polymorphisms, because there are a lot of more and more examples of that. A good one is there are people who are resistant to HIV infection, and it's thought that that gene that has a mutation in it might have conferred resistance to plague, and it might have been selected for in the population because of plague resistance, and a bystander effect is HIV resistance, because HIV entered the human population really recently.
Sure. Anyone else? Okay. And yeah.
I should like to mention that one thing when we talk about evolution of traits or evolution of organisms is that one tends to think that things get better and better in some sense, and certainly this is not how evolution works. So there are always trade-offs, and this is what Ted and Susan was talking about with respect to balancing polymorphism. There are trade-offs in which traits that are good in one context can be bad in other contexts. In fact, some people think aging in of itself, because we know many organisms don't quite show the drastic aging traits that we do, is also a kind of evolutionary adaptation where certain genes that are good for you when you're young are selected for, and then they tend to be bad when you're old, because maybe the things that were good to useful metabolism is bad for old metabolism. So nature is always in balance, and evolution is really about following and tracking this balance of nature, not about things constantly becoming better and better.
Thank you. No, that's a good point, Dr. Kim. And actually, I should have done this earlier, but maybe I should ask any of you to sort of tell us for those of us who may not be familiar, what is a polymorphism?
Well, I mean, a polymorphism, if we're talking about a genetic polymorphism, is simply a variable site. Variable site in the genome. That's the simplest answer, I think.
So we might, in the vernacular, so to speak, talk about it as mutation, or as a difference in a couple of base pairs. That's probably a little too fancy, but.
Right. Let me also, mutation is somewhat of a difficult word, because what mutation implies is something abnormal in the vernacular, but actually what we mean in genetics is it's just something different from what we had already known before. So by polymorphism, what really we mean is if we look at a particular gene or a particular letter in the genome, that there's multiple variations that we see in the population. So there are certain sites we actually do know are completely identical for everybody, and that's what we talked about with 99.7% identity, that if we look at most of the letters of our genome, everybody has the same letter. But then there are about 10 million places where people have different letters, and that's what we mean by polymorphism.
Okay. Dr. Ross, there's a question I think someone picked up on something in your bio. What are arena viruses? Is that something about football or gladiators?
So for those of you who know Latin or even Spanish, arena means sand. And when you look at these viruses under the microscope, they have a sandy appearance, and so that's where they get their name. So the viruses that we study are endemic in South America, and they cause something called hemorrhagic fever, which is basically a bleeding disease. So that's really what they are.
Okay. There are a couple questions here that sort of, I think, go together. At least I'm putting them together, so that now they go together. And they're about kind of the, if you will, the potential promise of the Genome Project and that sort of thing. And I'm going to, again, lump them together and feel free to answer as you wish. But, and this is sort of a specific question, I think, for Dr. Kim, but the first one is, how much does it cost at this point to sequence a human genome? We hear a lot, one person's genome. We hear a lot about this sort of $1,000 genome that everyone's talking about. I saw someone's slide had a 454 on it. How much, what does that cost?
So right now, the cost is, it depends a little bit on the accuracy at which we assess a genome. So because of the techniques that we use, we can't just take a single snapshot of the genome. What we do is it's like taking multiple X-ray pictures. And so if you take more and more pictures, you get a more accurate assessment. So to the degree of accuracy we generally like to look at, it costs about somewhere between $10,000 to $15,000 per human genome right now. And that's remarkable compared to the about $2 billion that it cost to complete the human genome in 1993.
And that number, I assume, obviously, from what you just said, is doing this. It's coming down.
It absolutely is. So we have been talking about $1,000 genome for some time, and I'll be willing to bet considerable sum of $5 maybe that within 5 years.
I think $1,000 would be the appropriate bet.
We'll sequence your genome, Ivan.
Oh, well, my insurance company will be very happy to hear that. And sort of a related question, and really for anyone, how has this knowledge that we've gained and the increasing ability to sequence, how has it informed the drug development process? This is obviously of intense interest to to companies right here in this area.
I think the benefits are just starting to be felt. The drug development cycle takes something like, I don't know, 10, 20 years, $1 billion to bring a new drug to market. So the first draft of the human genome was only completed in 2001. But there are many ways that it will benefit health going forward. To just pick one example, humans metabolize drugs quite differently from each other. Some people will get rid of a drug quite quickly and might need a higher dose of a drug. Others might make some toxic substance and harm themselves when they're exposed to some drug or other. Some might have an immune reaction, anaphylactic shock. So there's very considerable hope, and to some degree a reality, of being able to look at human DNA and figure out in advance how they may respond to different drugs, and thereby tune the therapy to make it as helpful as possible and minimize the harm that could be done. And so that's just one of many, many examples.
So the big catchphrase now is personalized medicine. And the idea is that if we can sequence your genome or some set of genes in your genome, then we'll be able to develop specific therapies that'll be good for whatever disease you have. And that therapy will be different from the person sitting next to you who has a different disease because their genes are different, basically. And it's really been done for a long time. I mean, we've known you can test people by looking at the proteins, and the big move forward now is we can actually look at the actual DNA sequence and begin to make predictions about how each individual will react to different drug therapies.
Yeah, I was just going to say that, that we're at the point now where we can be talking about population genomics. We can actually be sequencing populations. And there's a project called the 1,000 Genome Project, some of you may have heard about, where that's exactly what they're doing. So they're at the stage of, they've sequenced at least 1,000 individuals. And so when we're looking at some of these common traits in Africans, for example, if I could sequence the entire genome, I may actually be able to find specific variants that play a role in these traits. But I did want to point out that even if miraculously we could sequence the genomes of every one of us in this room, we're a long ways from understanding the effect of that on these complex traits and in terms of developing more effective therapeutics. So we're going to need people like Dr. Kim, who are computational biologists, and we have to better understand the sort of translation from the genetic variation to the actual variable trait of interest or drug metabolism.
I actually have a question, and again, maybe Dr. Bushman, but anyone really to answer. There was a lot of interest, and there still continues to be, although slightly differently, in the past several years and further back about not just microbes that sort of cause diseases we think of as infectious, the flu, HIV, et cetera, but actually diseases we don't think of as infectious. I'm thinking in particular of whether or not certain infections may give us a higher rate of heart disease, things like that, hardening of the arteries. Is there some relationship to inflammation? What's in a sort of broad sense, where is that field? What is interesting going on there now?
Well, how much time you got? There's a ton of interest in these kinds of questions, and people are now that we can map bacterial microbial communities using the deep sequencing, we're building up gigantic data sets over just the kinds of samples you were saying, arterial plaques or inflamed tissue from inflammatory bowel disease or whatever, to try to see how the microbial populations are altered and try to associate characteristics of the community with the disease state. This may be a case where there's not like one bug doing something bad and making you sick, but it may be a whole population of microbes changing in some ensemble kind of way into a dysbiosis, into a community that's harmful as a whole rather than just some individual bad actor. But then the deep sequencing methods, the new approaches are also allowing people increasingly to identify new bad actors that they didn't know were there before. So there have been a whole bunch of studies now, wonderful examples of people taking samples from subjects who had the outward signs of some infection, but they couldn't find any of the usual bugs. Purifying, collecting clinical samples, purifying DNA, deep sequencing, and identifying completely new bugs that they didn't know were bad, but now do know that they're bad. And so the field of virology, for example, has really been upended by the finding of a whole bunch of interesting new viruses by this route, for example.
Great. Anyone else on that?
Just to play the devil's advocate, as difficult as it's going to be to connect polymorphisms in the genome to particular ability to metabolize drugs, the big problem with infectious disease is proving that an infectious agent, whether it's a virus or a bacteria, really causes the disease. And so that's a big challenge because we don't intentionally infect people with viruses and show that they come down with a disease. So there'll be correlations, but it's going to be difficult to prove a lot of these correlations.
Absolutely.
Although this doesn't necessarily rely on the deep sequencing or the knowledge of new viruses, part of what is also emerging from the use of genetic methods is, in fact, how widespread different forms of bacteria and viruses have been brought to different parts of the world. And I'm thinking in particular the Americas. So, for example, different kinds of viruses like human papillomavirus are causes of cervical cancer. And some of the studies we're doing and elsewhere are looking at the transmission of these viruses and where they happen to come from. There are some that have certain geographic origins in Europe or Africa or other places, and so you can actually identify where they've come from and being brought into indigenous communities because of contact between those or admixture between those communities. Likewise, Helicobacter that causes pylori that causes gastric ulcers are being widely studied across the north in indigenous communities, and most of those are coming from contact with Europeans over the past few centuries. So what we can get some sort of insights into is sort of this emerging global epidemiological environment whereby we can track the movements of these particular pathogens, which are not necessarily infectious agents per se, but do cause chronic disease and cancers, and try to trace their transmission and get a handle on the virulent forms that happen to be present in the case of HPV, or in fact ways of trying to mitigate those diseases in different communities.
And that's sort of a related question, which is if we're looking at all of these microbes and what they're doing, we're looking at humans and what effects they have on humans. And maybe Dr. Kim, if you could take this one, what percentage of and there's obviously no microbial genome unless you think about just the world as a great big genome, but what percentage, say, of important microbes have been sequenced, or what's not been done yet?
That's a great question, and so there has been systematic efforts to sort of catalog all known organisms, and of course before we would just go out and see what's there, and now we actually look at things we can't see and just sequence and find out what's there. So there's about 1.5 million sort of described organisms in the world. And so if you sort of go and ask a systematist or taxonomist, they'd be able to name 1.5 million, not a single person, but in collection. There used to be estimates that maybe the total biodiversity was maybe 10 times that we didn't know about, so we knew about 10%. Now with all of this deep sequencing and all of this is coming out, we think maybe we know about 1%. So there might be, say, 100 million organisms that we know for which we are just beginning to see some evidence of, mostly through these kind of DNA sequencing.
Great. Thank you.
I think it's very hard to estimate. The richest bacterial environments, I'm told by colleagues in marine biology, may be deep sea sediments, the mud at the bottom of the ocean. So if that's true, people haven't spent a lot of time trying to study what kind of bugs there are in the mud at the bottom of the ocean. So the potential for huge sources of biological diversity that we've barely looked at all is, I think, really high.
Absolutely. Thank you. We talked about personalized medicine. Dr. Ross, you mentioned that, and we're sort of thinking about the drug development process. Very specifically, how does a lot of this knowledge, how does it contribute to the future of gene therapy? Are there things that we're learning in particular that might help in terms of actually coming up with successful gene therapies, or are there some genetic therapies already that have been out there?
So I actually serve on the Recombinant Advisory Committee to the NIH, which is the committee that basically oversees all gene therapy in the United States. So there are lots of diseases now that are being treated with gene therapy. Probably the most successful diseases are genes where humans have a genetic defect that can be corrected by correcting their blood cells, basically. So if they're missing an enzyme or something, there are ways to introduce the gene into the blood cells and correct the genetic defect. However, I can tell you that there are as people learn more about what genes are involved in cancer or in developing ways to to treat cancers, more and more therapies are being tried where people are trying to engineer human cells so that they'll fight off cancer better, so that your immune system works better, and so that we can specifically target cancer cells. So that's probably the major area for gene therapy besides single gene defects where the blood cells can be corrected. So the more we learn about the genes involved, the more we can use gene therapy to correct those diseases.
I wanted to ask, and again, I think this is sort of a question for Dr. Tishkoff, but sort of an ethical question, if you will. One of the members of the audience asked, you're obviously collecting a great deal of information, genetic information, characteristics, various things, measurements, and what have you. What happens to that information? Obviously, you publish on the information. What happens to it? How does it get fed back into that community? And could a commercial venture, or anyone for that matter, have access to it, and under what circumstances, under what provisos?
So most of this information, our purpose is to use it for academic research, and so we will publish the results, and hopefully that will, benefit both the academic community, and we hope also the people who contributed the samples as well. But my group, we personally believe that it's critical to return results to the individuals who contributed. And just recently, I was in January, I went back to Tanzania and visited most of the tribes that had contributed to the studies and actually translated the main take-home points of the studies and brought them copies of the science paper. And in fact, and they loved it. I mean, they were absolutely thrilled. And my student, I have a graduate student from Kenya, and he went back to every population in Kenya and brought them back the results. And again, they were so happy. And there was, at least one group we studied, the Sandawe hunter-gatherers, where I remember when I showed up there, they said, "Oh, are you coming back to tell us our results?" Because somebody had gone there, I think in the 1940s or 1950s, and they knew the exact date. They remembered exactly when that person came. And they were studying classic blood polymorphisms, and nobody ever came back to tell them the results. And so they were really thrilled to be participating in this, and they're particularly interested in the information about ancestry and so on. And in terms of the commercial use of this, it's been a particular concern of mine that, of course, when the data gets published, it goes into the public domain, and it really can be used by anyone. It can be used by commercial endeavors and so on. And so it's a matter of debate whether one should try to regulate that, for example, by using patents. Because one possible way to do that is if you do patent this, if you have the permission of the populations, any profits can potentially be returned back to the populations. And it may actually be a way of benefit sharing with the groups.
What Sarah's describing is happening in many places, actually involving human population studies, that there has been really a shameful practice of not returning results and basically engaging the communities with the findings of the studies, and actually enlightening them on the genetic data, but also basically developing a discourse or a dialogue about the results, and developing, I guess, a level of engagement which actually is mutually enriching and allows the researcher to learn as much about the community as the communities do from the researchers. And so it's our practice to do much the same as Sarah's describing, actually co-publishing with indigenous authors or community people who are involved in the research with us as partners. And also taking as many steps as possible to make sure that the results that are published are anonymous, that the identifying information can't be used in any ways that would be not at the discretion of the participants themselves, because of concerns that Sarah's describing. But I think we're, as molecular anthropologists or as population geneticists, we're working within a new sort of climate of where there's an explicit attempt to work with communities in all ways possible, and including the return of results and working together to describe the findings. Now, that may not be possible in every case because of the technical nature of the data, but I think this is really a sea change from practices of the past.
Great, thank you. And we're coming close to closing, and I know that there are a number of young people in the audience. We saw two of them actually in slides. One of them, if she hasn't eaten the candy yet, I think will be soon. And so I wanted to actually sort of give the final question to them a little bit, and I understand there's a high school class here as well. And so what I'm going to do is sort of a lightning round, if you'll forgive me that, which is neither Stewart nor O'Reilly. I think it's more ESPN. And ask you each to, given your particular interests and what you study, to do two things. One is, and this is from the audience, what is your favorite microbe, or what is your favorite gene? You can only choose one. That's the very explicit instructions on this card here. And the other, a little more seriously, is for those young people in the audience, what would you recommend if there were, and obviously if they're here tonight, they're interested in what you do and in pursuing it potentially, what would you recommend for them if they were someone in high school now? Are there opportunities they can take advantage of?
Okay, I don't know. I just have lactose on my mind. That's one of my favorite genes. I'm sorry. So I'm going to pick that as one of my favorites. But in terms of what you can do, we actually have high school interns in my lab, and there's a number of faculty at Penn who take high school interns. And so the best thing you can do is basically contact us, and sometimes people have written to the chairs of departments, and the chairs will send out a notice, or you look us up on the website and you contact us individually. And if we have openings in our laboratories, you actually can come in after school and get some first-hand training. And that really is one of the best ways, I think, to learn the research methodology.
Favorite microbe, I've got to go with bacterial viruses, like in that movie, The Lunar Lander, like pinning the bacteria. Those guys are just really cool, I think. So for kids, I guess I would suggest try to learn a lot of different things, study lots of things in school and find what you're excited about and follow that. But at a relatively young age, it's fine to start to go deep into things, but learn widely. Try to find things that really excite you to follow up on. Science worked for me in that, but you need to find what works for you.
As a genomicist, I think I'd be remiss if I said there was a favorite gene, so I'd have to go with the whole genome. As far as for the students, I reiterate what Sarah said, that you can get involved very early, and I'd encourage you to get involved very early, with one caveat, that is that we showed a lot of fancy slides and gave interesting stories, but science actually is extremely tedious. It takes long and repetitive work, and so you have to be persistent. And so if you do volunteer and you do get involved in research, be persistent and don't give up, because it takes a long time until that real reward comes to you.
So I'd have to say retroviruses like HIV are my favorite pathogenic organism, or pathogen. But not only because they cause disease, but because, as Rick mentioned, they're in our genome and they cause genetic diversity, and also because they're used as gene therapy vectors. So they're really interesting from their ability to cause disease and their ability to cure disease as well. I also have always had high school students working in my lab. I'm really proud to say one of my former students was a finalist in the Westinghouse Fair, or whatever it's called now, I forgot the name of it, and is now a graduate student at Penn in immunology. So I think many scientists like training students at a young age, and so I urge you to seek out mentors. There's lots in the Philadelphia area, not just at Penn, but at a lot of the institutions in Philadelphia.
Dr. Schurr, you have the last word here from the panel.
Okay, well, I would say my favorite part of the genome is the mitochondrial DNA, which I've been working with for 20-some years. It's a neat little piece of the DNA. It's only 16,569 base pairs, a trifling amount compared to the whole genome, but it's absolutely essential for life.
So that everyone knows, the mitochondria is?
The mitochondria are basically organelles within the cells, or little functional entities, which are basically the so-called powerhouse of the cell. They generate all the cellular energy that basically keeps us running, and they're billions of years old. Symbiosis with eukaryotic cells, or with cytoplasmic-like organisms, gave rise to eukaryotic cells, and then to a variety of different kinds of life forms. So they're integral to all aspects of life. Involved in disease, involved in normal metabolism, probably in adaptation, and a whole variety of other sorts of things like that. So they're a neat thing to study, and have a very complex interaction with the nuclear genome as well, since they gave up a number of their genes to the nuclear genome to be able to be in an integral association with that particular cytoplasmic organism. So there are a lot of complexities and interesting details about the mitochondrial genome that one can see from looking at those genes. So that's sort of a long answer to a short question, but in any case, from a high school standpoint, I would agree with everything that's been said here, and I would only reiterate that another way of getting some insights into what you might want to do in the future is to go to professional meetings. I think at the AAAS and other places, sometimes at the anthropological meetings, there are sessions where students can actually attend and even present their own work. And all of us tend to would like to encourage that as much as possible. And also welcome the contact of high school students if you're interested in working in our laboratory. So I think it is an important way of getting a foot in the door and understanding what you'd like to do by getting your hands wet, literally and figuratively. So do take some, venture out and contact us and try to find something that interests you.
Terrific, thank you. And with that, speaking in fact of education and outreach, I want to introduce Dr. Kristin Field, who's the Associate Director of PGFI, and she actually is quite involved in the outreach and what have you. She's also interested in diversity and equity issues in science. And she's worked at Monell here in Philadelphia and has a PhD in ecology, evolution, and organismal biology from the Ohio State University. Dr. Field.
Okay, well, this will be really short because these are just the closing remarks, and everyone's been very, it's been wonderful to have everyone here. The turnout tonight has been really heartening, especially since this program has had much less of an obvious medical focus than our previous ones. Genomics clearly has great promise for applications towards medicine and solving societal problems, but it also, as you heard a lot about tonight, is revolutionizing how we can approach many of the fundamental questions about how the world works and how, as humans, we fit into this world. So support for research on fundamental questions can be lukewarm from the public and from critical funding sources. So we really want to thank you on behalf of the whole PGFI for coming out tonight and showing your support for programs like this. I would also want to give a huge thank you to all of the speakers and panelists and to Dr. Oransky for his able moderation. All of them have volunteered their time to share their expertise and thoughts tonight. Finally, many thanks to our Franklin Institute partner, in particular Karen Alenich and Dr. Frederic Bushman. This is our 4th event with the Franklin Institute, and we try to organize one every spring and in the fall. A recording of this event will be available on the PGFI website, along with the recordings of past events. In your program, you will find the PGFI website web link. So also in your program, you'll find a survey, and I hope you will take a few moments to fill it out. This is really important to us. We use the feedback to make each program better. And also, if you want to be notified of our upcoming programs, please leave your contact information on the survey. There's a dropout box in the back. Again, thank you very much for coming out, and we wish you a great evening, the rest of the evening.